This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
It's my pleasure to introduce Chelsea to present her completion seminar. As you can see from the title, she's presenting on granulomatous colitis in dogs and looking at evaluation of inflammatory markers, goblet cells, and MUC2 RNA expression. Before we start, I'd just really like to congratulate Chelsea on getting to this stage of her completion. She's had some incredible challenges that have been quite formidable to overcome during this master's that have been completely beyond her control, and she's done an excellent job in overcoming them. So I'm looking forward to this presentation. Thanks, Chelsea. So Chelsea, you're still muted.
All right, we'll try again. Okay, can everyone see the slide? All right, good afternoon, everyone. Thanks for listening in. I am excited to share with you the details of my research master's project, which involve evaluating Ki67, goblet cell, and MUC2 RNA expression in canine granulomatous colitis. I'll begin by outlining the background to my study, research objectives, followed by discussing materials and methods, and then summarize study results, followed by key discussion points and conclusions. The gastrointestinal tract is the most dynamic immunological environment in the body, with constant exposure to thousands of dietary antigens, commensals, and pathogenic microbes. The three key components of gastrointestinal immunity are outlined in this diagram. My project focuses on the intestinal mucous barrier and the innate immune system. The innate immune system is effectively the first line of defense and is composed of various sentinel cells which recognize PAMs, or pathogen-associated molecular patterns in the gastrointestinal lumen, which collectively trigger rapid responses against these antigens. Important sentinel cell types include intestinal epithelial cells, which form an effective physical barrier against pathogens, as well as goblet cells. The additional roles of innate immunity include initiation of adaptive immunity and orchestrating tolerogenic immune responses. Intestinal mucus is a viscous hydrated gel from mucin glycoproteins and exists as an inner and outer layer in varying proportions across the gastrointestinal tract as seen here. It also plays numerous essential roles as outlined. MUC2 is the predominant gel-forming mucin in the small and large intestine and forms stratified layers as the building block of intestinal mucus, seen here, therefore functioning as a selectively permeable filter preventing bacterial entry. It also has resistance against proteolytic activity and has a role in limiting immunogenicity of gastrointestinal antigens. Goblet cells are the most abundant in the small intestine and upper colonic crypts. They are the primary source of mucins and contain mucus-filled membrane brown granules as seen here. Goblet cells are responsible for mucus secretion, which usually occurs at basal levels by sentinel goblet cells. However, they can also occur rapidly during inflammatory or infectious stimuli by peripheral goblet cells. Goblet cells require robust endoplasmic reticulum function due to their role in mucin production, which are large complex molecules containing numerous disulfide bonds. In people, colonic inflammation leads to increased mucin biosynthesis by goblet cells to match increased requirements. The excessive workload on goblet cells renders them susceptible to damage and apoptosis, as well as inappropriate folding of MUC2 molecules. These events result in a thinner, poorer quality mucus layer, which mucolytic bacteria can more easily penetrate to incite an inflammatory response. The presence of enteropathogens, toxins, and inflammatory cytokines collectively result in a series of events, including MUC2 depletion, alterations in mucus composition leading to poor quality mucus, mucus degradation by bacterial enzymes, and intestinal dysbiosis. Consequences include breakdown of the mucus barrier with subsequent translocation of pathogenic and opportunistic bacteria, and the persistent contact between these invading bacteria and the gastrointestinal epithelium activates submucosal immunity and perpetuates inflammation, which is a key event in the development of many chronic inflammatory enteropathies. Moving on to granulomatous colitis. This is a form of canine inflammatory bowel disease, or IBD, which parallels ileal Crohn's disease in people. It predominantly affects young boxers and French bulldogs less than 4 years of age worldwide, with associated clinical signs including that of colitis, specifically frequent small volume diarrhea, tenesmus, mucoid feces, hematochezia, but can also be fairly non-specific, ranging from lethargy to cachexia in more severe cases. Disease resolution is possible with age. Both host and pathogen factors contribute to the development of GC. Host factors encompass that of breed-specific genetic aberrations, specifically that in boxers and French bulldogs, a dysregulated host immune response, and overall ineffective microbial clearance. Pathogen factors refer to specific virulence characteristics of mucosally invasive bacteria, not typically found in health, in particular adherent and invasive E. coli. These bacteria possess the ability to infiltrate gastrointestinal mucosa and survive within macrophages to incite and perpetuate granulomatous inflammation. GC usually confers no significant findings on routine screening, which include that of blood work and abdominal ultrasound. Gross abnormalities on colonoscopy typically include that of a radon, cobblestone, ulcerated colon mucosa, as seen from the endoscopic image here, which was from one of our patients seen at UVet Hospital in 2018. Colonic biopsies are also subject to histopathological staining. Routine H&E is used to highlight histiocytic inflammation, and periodic acid shift or PAS staining is used to highlight PAS-positive macrophages, which is a key feature of this disease. Bacterial culture and susceptibility can also be performed, and for identification of mucosally invasive bacteria, specifically adherent invasive E. coli, fluorescent in-situ hybridisation of fish is often used. On routine histology, classic findings with GC include that of goblet cell depletion in the colonic epithelium, histiocytic inflammation, marked colonic mucosa ulceration, and infiltration with PAS-positive foamy macrophages as seen in this magnified window. Additionally, as mentioned earlier, FISH is a tool which utilizes different colored probes to highlight the presence of pathogens in tissue biopsies to corroborate with the diagnosis of GC. Image A represents that of healthy canine colon, where the microflora identified in red is confined to superficial mucus and glands. The blue cells you see here are the colonic epithelial cells, and these regions form the crypts of the colonic crypts. Image B represents that of a colonic biopsy in a dog diagnosed with GC, where you can see that bacterial infiltration of colonic mucosa can be appreciated in red here, and that's consistent with adherent invasive E. coli within PAS-positive macrophages.
Yeah, what is it? What does it mean?
Ki67 antigen is a marker of cell proliferation and is expressed in all active phases of the cell cycle. It has been evaluated as a marker of intestinal inflammation in both people and dogs with chronic inflammatory enteropathies, as highlighted in the following publications below. However, the utility of Ki67 has not yet been evaluated in dogs with GC and lymphoplasmacytic colitis at this point. Colonic goblet cell depletion has been subjectively observed in dogs with GC and non-GC colitis, where more severe goblet cell depletions were observed in ulcerated colonic mucosa, and in GC dogs, these goblet cells were replaced with inflammatory cells and clumps of cocobacilli consistent with adherent invasive E. coli. Another publication noted that substantial increases in goblet cell numbers in post-treatment biopsies of GC dogs occurred in conjunction with eradication of adherent and invasive E. coli. However, these studies have been based on subjective observations rather than quantitative assessment of goblet cells. And one key reason is due to the inherent limited ability to accurately assess mucus and goblet cell populations in intestinal tissue using routine tissue preservation techniques. As such, goblet cell and MARC2 expression have not been quantitatively evaluated in canine colitis to date. With that, the key objectives of my study were first, to compare Ki67 expression levels amongst dogs with GC, lymphoplasmacytic colitis or LPC, and non-colitis dogs using immunohistochemistry, to compare goblet cell and MARC2 RNA expression levels as indirect markers of mucous barrier health in dogs with GC, LPC, and non-inflamed colon using semi-quantitative methods. And finally, to explore the use of RNAscope in-situ hybridisation for evaluating MARC2 RNA expression in both healthy and diseased canine colon.
Awesome, thank you.
Endoscopic colonic biopsies from a 7-year period were retrospectively and prospectively recruited for this study. A total of 48 dogs were classified into 3 study groups based on their final histologic diagnosis. Non-colitis dogs were dogs that did not exhibit colonic signs and had no to minimal colonic inflammation identified on histology. These dogs had undergone colonoscopy for further investigation of small intestinal signs in a separate clinical trial. Biopsy samples were then reviewed by a board-certified pathologist with grading of colonic inflammation, standardized using the World Small Animal Veterinary Association or WASAVA histopathological guidelines. The histologic score for each biopsy section was derived from the sum of 8 histologic parameters as outlined in the table here. The cumulative score for each section ranged from 0 to 24. Fresh slide recuts per dog were then subject to 2 histological staining techniques. First, Ki67 immunohistochemistry was performed. The immunolabeling procedure involved using a monoclonal mouse anti-Ki67 primary antibody mix, which has been previously validated for use in dogs. Ki67 positive or immunoreactive cells are highlighted in red, as seen in the image on the left. The second technique was to perform Elsing blue periodic acid shift or ADPAS staining to highlight colonic goblet cells, which appear as an indigo blue due to their acid mucopolysaccharide component, as seen in the image on the right. Subsequently, MARC2 RNA localization was assessed with RNAscope in-situ hybridisation, which is a novel technology, utilizing customized canine MARC2 targeted probes and an amplification system to quantify MARC2 RNA present in the colonic epithelium. MARC2 RNA signals were then visualized with standard light microscopy as brown stains within goblet cells, as seen from the image below. For each hybridization run, the housekeeping gene CLUBC was used as the positive control, while DAB, which is a bacterial gene not expressed in mammalian tissues, served as a negative control probe. Due to limited research funding and reagents, only the best quality biopsy samples were selected for the RNAscope assay. And this included a total of 21 dogs, 8 GC, 7 LPC, and 6 NC dogs. Following cell staining, 5 representative microscopic fields per dog containing longitudinally oriented crypts were then selected by a single observer, myself, from digitally scanned slides. These fields were then analyzed using Fiji, which is a computer image analysis program, to assess 3 variables. The first being the Ki67 labeling index, the second being a goblet cell staining percentage, and the third being the MARC2 index. Normalized MARC2 index and Ki67 index results were generated by dividing the result by the respective WASAVA histologic score for each dog. As the semi-quantitative assessment of these 2 variables involved counting the number of nucleated cells per field, normalization of these variables aimed to account for the severity of inflammatory cell infiltrates, which could artificially lower the labeling percentages per field by a dilution effect. Using the Fiji image analysis program, automated cell counts were generated using a customized algorithm. This involved manually selecting the regions of interest for analysis, as seen from the yellow circles here, if you can see closely, and generating individual color channels to highlight different components, such as the lamina propria versus the epithelium. An isolation of cell nuclei was achieved associated with a positive nuclear signal was performed for quantification. The images on the right depict the histologic and RNAscope images of selected microscopic fields, along with their paired Fiji images. Ki67 positive cells are highlighted with immunohistochemistry as red, as seen on image A, and green with Fiji on image B. Goblet cells are visualized as indigo blue with the APPES stain in image C, and green-yellow with Fiji on image D. And lastly, for goblet cells with MARC2 positive uptake, visualized as brown using the RNAscope, as seen on image E, and green with Fiji seen from image F. Following which, the distribution of cell count data was evaluated for normality using the Shapiro-Wilk test, and descriptive findings were outlined using box plots, and Pearson correlation coefficient was used to evaluate the associations between variables of interest. Finally, 3 linear regression models were performed to assess first if WASAVA histologic score significantly predicted 3 outcome variables, as mentioned earlier, and if goblet cell staining percentage significantly predicted MARC2 index. This involved using interaction terms and comparing the magnitude of differences, gradients of trends, and their 95% confidence intervals amongst the 3 study groups. Table and graph above outline the signalment of the sample population for each study group. Unsurprisingly, the GC group comprised of only young boxers and French bulldogs, predominantly less than 3 years of age. There were no age or breed predispositions noted in the other 2 study groups. Descriptive results comparing semi-quantitative parameters among study groups are outlined using the box plots, box and whisker plots. On the left, you can see that GC dogs had the most severe WASAVA histologic score, with a median score of 10, and that was approximately 2.5 times that of LPC dogs and about 6.7 times that of NC dogs. On the right, median normalized Ki67 index was highest in non-colitis dogs, which was approximately 6.6 times that of the GC dogs and about 1.4 times higher than that of the LPC dogs. So overall, the Ki67 results indicate a higher cellular proliferation rate in non-colitis dogs, followed by LPC and then GC dogs. These results were surprising as they differed from previous publication results, which have observed upregulated Ki67 expression in dogs with inflammatory enteropathies. Both reasons for these observations will be elaborated later. Additionally, seen in the left box plot, median goblet cell staining percentages were the highest in GC dogs, followed by LPC dogs and then NC dogs. On the right, median normalized MVS index was the highest in non-colitis dogs at approximately 10%, which was twofold higher than that of LPC dogs and threefold that of GC dogs. We can see here that interestingly in GC dogs, although greater goblet cell numbers were observed, MVS expression appeared significantly lower compared to the other study groups, suggesting a degree of goblet cell dysfunction. Key results from the correlations between selected variables are represented by scatter plots on the top left and the paired regression model data on the bottom right table. For each slide, subsequently, the results of interest are in the last 3 rows of the table, which are highlighted in the red box. These reflect the gradients of trends in the outcome variable. In this case would be the normalized Ki67 index, with every unit increase in the explanatory variable. In this case, the Wasaba histologic score per study group. And if results for the LPC group and NC groups were significantly different from that of the GC group, which has been set as the reference group. When assessing the relationships between Wasaba histologic score and normalized Ki67 index, observations included a strong negative correlation in the non-colitis dogs, as seen from the downward sloping orange line. And this correlated to approximately a 2.47% decrease in Ki67 expression for every unit increase in Wasaba histologic score. In comparison, the moderately negative correlation was noted in the LPC group, as noted by the gray line here, and that corresponded to a statistically significant decrease in Ki67 expression of 0.87% per unit increase in Wasaba histologic score. And lastly, a weak negative correlation was noted in the GC group, as portrayed from the flat blue line here, and that corresponded to a weak, non-statistically significant decrease in Ki67 expression, with increases in Wasaba histologic score. Next, when evaluating associations between Wasaba histologic score and goblet cell staining percentages, weak negative correlations were observed in both LPC and non-colitis dogs. And you can see from the linear regression data, for every unit increase in Wasaba histologic score, a 1.09% decrease in goblet cell expression was noted in LPC dogs and a 1.16% decrease in non-colitis dogs. That corresponds to the orange line here for the non-colitis dogs. In comparison, the lack of correlation was noted in GC dogs, as supported by the flat blue line here, and minimal changes, about 0.02% decrease in goblet cell expression for each unit increase in Wasaba histologic score. In summary, although GC dogs had the most abundant goblet cell populations amongst the 3 study groups, minimal quantitative changes in goblet cells occur in the face of colonic inflammation. In contrast, moderate goblet cell depletion was observed in LPC dogs with increasing colonic inflammation, which likely reflects goblet cell injury and apoptosis due to excessive workload. The third association evaluated was between Wasaba histologic score and a normalized MVS index, which revealed a strong negative correlation in the LPC group, supported by the gray line on the left scatter plot, and a 2.6 decrease in MVS expression for every unit increase in Wasaba histologic score. Interestingly, in contrast, weak negative correlations were observed in GC dogs, seen from the blue line here, and a 0.34 decrease in percent decrease in MUC2 expression. And for non-colitis dogs, with a 1.9% decrease in MUC2 expression, and for every unit increase in Wasava histologic score, and the gray line here. In summary, overall, the strongest magnitude of decrease in normalized MUC2 expression was observed in LPC dogs, which was, from the linear regression data, approximately 7.6 times that of GC dogs and about 1.4 times that of non-colitis dogs. When assessing the effect of individual histologic parameters on MUC2 expression, which was done on separate scatter plots, the magnitude of negative correlation in LPC dogs appeared to be influenced by the severity of superficial epithelial injury and crypt hyperplasia, which, if you recall, are two of the histologic parameters included in the Wasava histologic score. And these likely reflect damage to your MUC2 producing goblet cells. The final association evaluated was between goblet cell staining percentage and normalized MUC2 index. No correlation was noted in GC dogs, as reflected by minimal changes in MUC2 expression, with increased numbers of goblet cells, and by the flat blue line here. A weak positive correlation was noted in LPC dogs, supported by the gray line here, and approximately 0.48% increase in MUC2 expression for every percentage increase in goblet cell numbers. And lastly, a moderate positive correlation was noted in non-colitis dogs, with a 1.37% increase in MUC2 staining percentage, MUC2 index, for every percentage increase in goblet cell numbers, as seen from the orange, line here. Overall, none of these values were statistically significant. To summarize the key study observations, GC dogs had the most severe colonic inflammation histologically, as indicated by the Wasava histologic score. Ki67 expression was moderately downregulated in GC and LPC dogs with increasing colonic inflammation. As mentioned earlier, these findings were surprising as they differed from observations in previous studies, and possible reasons for discordant results include having a small sample population, only selecting 5 microscopic fields per dog, and our methodology, which included counting on nucleated cells in each field, which would have included cells, apart from mononuclear leukocytes that typically express Ki67 antigen. Notably, the increased number of inflammatory cell infiltrates in GC and LPC dogs would potentially have lowered the Ki67 index, as inflammatory cells do not typically proliferate. Ideally, we would have considered assessing the spatial distribution of Ki67 activity in our biopsies, for example, upper and lower crypts, lamella propria, colonic epithelium, but this fell outside the scope of our study. Additionally, Ki67 was markedly upregulated with increased MUC2 expression in the GC group, which may be in part due to enhanced MUC2 biosynthesis and hypersecretion by goblet cells. Goblet cell expression lacked significant correlation with severity of colonic inflammation in all study groups. And for MUC2 expression, this appeared moderately and significantly decreased in LPC and GC dogs respectively. With increased colonic inflammation, MUC2 expression sharply declined in LPC dogs, but remained relatively static in GC dogs. This might indicate direct damage to goblet cells within the colonic epithelium and potentially stem cells in the crypt base, which are responsible for goblet cell differentiation and renewal. A final point would be that goblet cell and MACTO expression did not appear to correlate significantly in GC and LPC dogs, suggesting that goblet cell metabolic activity rather than quantity increases to boost mucin production. The limitations of this study were largely due to its retrospective nature, as outlined here. Of note, FISH results were not available in most cases, as anecdotally, most clinicians do not typically submit biopsies for FISH, but obtain a diagnosis of GC from routine histology and potentially therapeutic response to antimicrobials. Similarly, post-treatment colonic biopsies are not routinely obtained in clinical practice to document resolution or improvement in histologic inflammation in dogs with colitis, as most clinicians rely on clinical response to guide therapeutic decisions. In conclusion, this study has provided an exciting opportunity to improve our understanding of goblet cells and mucin as components of the innate immune system in healthy dogs and changes which occur with chronic canine colitis. This is also the first study to explore the use of RNAscope to evaluate MACTO expression as a marker of mucus health in both healthy and diseased canine colon. The differences in MACTO expression between GC and LPC dogs likely reflect differences in the cellular pathways regulating MACTO biosynthesis and secretion, and specifically the loss of ability to produce and secrete MACTO by colonic goblet cells may be a key contributing event in the disruption of the mucus barrier in lymphoplasmacytic colitis. Lastly, no significant correlations were noted between goblet cell and MACTO expression in this study population, which would suggest that MACTO expression levels appear influenced by mechanisms modulating goblet cell function rather than quantity in these forms of canine colitis. Several avenues for future investigations include evaluating for inherent aberrations in MACTO gene expressions in canine breeds of individuals predisposed to colonic IBD. Comparison of goblet cell and MACTO expression in pre- and post-treatment biopsies will also be interesting to investigate. Specifically, it would be interesting to know if MACTO gene expression levels reach healthy levels post-treatment or improves to subnormal levels sufficient to maintain a disease-free state in these dogs. It would potentially be useful as well to assess the impact of long antimicrobial causes on the mucus barrier, particularly in GC dogs. The insight gleaned from this study will hopefully inspire future studies targeted towards improving current knowledge in the pathogenesis and management of canine chronic inflammatory enteropathies, including studies aimed at qualitative assessment of mucin structure and mucus layer components in canine colon, development of novel diagnostic biomarkers, and development of therapeutic strategies targeted towards boosting mucus barrier health, such as modification of mucin biosynthesis. With that, I would like to thank the following people who have been instrumental in my project these 3 years. The following are my references, and I'm happy to take any questions.
Thanks, Chelsea. That's a great presentation and summary. Does anybody have any questions? I'm going to take that as being no. I suspect that's because you did such an amazing job of summarizing everything.
Thanks. Well, thanks everyone for listening in. Yeah, any questions, let me know.
Great. Thank you very much again, Chelsea. Thank you.
That's all right. Thank you, everyone.
Bye.
Bye.